you didn't start the recording, go ahead. Sorry, trying to get oriented. So, welcome to Monday, a different kind of Monday, I think, for all of us. Today is Monday, March 30th, 2020. And today we are going to hear from Dr. Byron Adams about Charismatic Megafauna in Antarctica. We're excited to have this presentation for you all. These little science content presentations are designed for our new cohort of teachers for 2020, of which I think we have Elaine is on, and for alumni, just to kind of take a different perspective at the science in the Arctic and the Antarctic and give us something to focus on. So today's presentation, I'm going to let Byron share his screen, but when he's talking, if you have questions, go ahead and type them in the chat area. And of course, we can also, you can unmute yourself later on at the end of his presentation and put your questions there or ask them live. So with that, I'm going to turn this over to Byron.
Okay, thanks a bunch. Happy to talk about the charismatic megafauna of continental Antarctica. So what the outline for my presentation today, first I want to talk about what the McMurdo LTER is, the Long-Term Ecological Research Group. I'm going to talk about why anybody should care about what I'm talking about today. And then I'll introduce the charismatic megafauna of continental Antarctica. All right, so first of all, talking about the Long-Term Ecological Research Program. This is a project that is funded by the National Science Foundation, and it's supported by the United States Antarctic Program. And to your right here, you'll see this map that shows a whole bunch of these little orange dots. These are long-term ecological research sites. Most of them are found in North America, throughout North America, and here in Puerto Rico. There are some down here in Moorea, and there are a few of them here in the Northeast, but you'll see there are 4 of them there in the Arctic. And our project here is to do science and monitoring. So we monitor things and look at the response of these ecosystems to something like climate change, for example. So we take long-term measurements. Every year we go back and measure the same sorts of things. That's what we call monitoring. And then we also do science experiments. We'll call that just straight up science. These are funded 6 years at a time, each one of them. And we're charged with providing both short and long-term perspectives of how the ecosystems respond to climate-driven changes. And then the idea is that we synthesize our work across this entire network, right? So that the work that we do in Antarctica, here in McMurdo down there, you see there at the lower right part of your screen, that's us. The work that we do there, we try and synthesize our results across this entire network of ecologists and different ecosystems to try and see if we can get at fundamental principles of ecology, very fundamental. So we're charged with things like the movement of inorganic matter, the movement of organic matter, studies of populations, and we're charged with getting at primary production, photosynthesis, net primary productivity. We're charged with studying the patterns of disturbance and the patterns of ecological recovery and succession. We also look at land use and land cover change and human environment interaction. So those are the things that each one of these sites across the network are studying, including our sites. So in Antarctica, there are 2 long-term ecological research sites. There's one out here on the peninsula. Antarctica itself looks sort of like a pork chop, right? There's this big part in the middle, and then there's this arm that sticks out to the side. And on that arm is the Palmer Long-Term Ecological Research Site. And much of their work is ship-based. It's done from a ship. My buddy Oscar tells me that they sample an area that's the size of Oklahoma at about the speed that you ride your bicycle, right? And they cover all this area here, doing a lot of studies of plankton growth, dynamics, krill, that sort of stuff. And they also have some very extensive penguin studies there as well. So very marine-centered research there. Our site here is in the McMurdo Dry Valleys. So we're a very terrestrial site on the other side of the continent. And so I'm on the right-hand side here, I'm a biologist, but the team as a whole is trying to get at ecosystem change, how this ecosystem responds to climate-driven changes, but not just how the soil organisms respond, but how the entire ecosystem responds. And so in order to get at that, you have to, my best friends are biogeochemists and chemists, geochemists, hydrologists, ecohydrologists, glaciologists, meteorologists. We put all of our data together, right, to try and understand how this entire ecosystem is changing. And so big job, right? More than one person. So here's what our team looks like. We have quite a large team of principal investigators to get at this project. And so these are some of the most brilliant scientists in the world. And at this point, often when I'm talking to school groups or doing outreach types of things, at this point, like to point out that historically, the research in Antarctica has been done primarily by men. It's been a male-dominated endeavor until really the last 15 to 20 years. But I also like to point out that we have 3 women on our team here. This is the last iteration. We now have more than that. But Diane McKnight and Tina Takas Vesbach and Diana Wall are 3 of the brightest, most brilliant, and most decorated polar scientists you will find. And I feel really lucky to have them on my team, on our team, helping us out. Those times have changed, man. And it turns out that if you want to get science done and you want to do the best science possible, if you don't have brilliant women informing your research, then you're not going to get the best science done, period. And so I'm happy to say that that's changing, and it's changing for the better, and it's changing quite rapidly. So this is what our ecosystem looks like, and some people might think that LTR stands for lazy times, easy research. Not the case. LTER stands for long-term ecological research. This is what the landscape in the McMurdo Dry Valleys looks like. So you've got the most dominant landform here is the soils, but you've got these glaciers, these hanging alpine glaciers. Those glaciers melt and they form streams. The streams flow periodically for a few weeks out of the year into these lakes. The lakes are permanently ice covered with seasonal moats that form around them. So we've divided our team up into different groups. We have the C504 team that studies glaciers. We have C507 and 509 that study streams. C508, which studies the moats, and C505 and 511 study the lakes, and 507 studies the soils. And the reason that 507 shows up in the streams and it also shows up in the moats is because we're of that landscape that connects the glaciers to the streams and to the lakes. And so our research actually overlaps or integrates really well with these other teams. So, whatever, why should I care about this? It seems really lame. Well, soil ecosystems, as it turns out, provide essential services for sustaining human life on planet Earth. And as it turns out, if you're down with things like clean water or fertile soils or biological control of pest organisms in agricultural ecosystems, if you think it's important to store carbon, if you think that the decomposition and cycling of nutrients is important, then you better be down with soil ecosystems because they provide all of those essential services. And so in order to better understand these, we need to better understand soil ecosystems. So that's why I think everyone should care about this. Yeah, whatever, you know, like I got dirt in my backyard, man. Why should I pay somebody to go all the way to Antarctica to study a bunch of dirt? Well, in Antarctica, we have very simple biotic communities. Mother Nature has done this terrific experiment by removing all the plants from above ground, because plants take stuff from the atmosphere and they pump it into the soils, and that complicates things, right? So Mother Nature's removed all the plants from above the soils, above ground, and then it's also reduced biodiversity. If you were to take a handful of soil from your home in Alaska or in Colorado or Indiana, and you held that soil in your hand, that handful of soil would have hundreds of thousands of animals in it, and maybe even 20 to 30 species of nematodes alone, right? And so those, if you wanted to study how your experiments influence the composition of that handful of animals, that would be really complicated to figure out how each one influences the other. In Antarctica, you just have a few species. And so we can do manipulative experiments, and we can identify how every single species responds to our experimental treatments, for example. And so for the same reasons that scientists studying developmental phenomena study like lab rats or nematode worms like C. elegans or sea urchin embryos and things like that, they pick their model organisms. Mother Nature has made this really cool modern model ecosystem for us to study that is simplified in ways that allow us to get at problems that otherwise might be too complex or untractable. These are really sensitive to climate variation, right? So life exists where you have liquid water in this ecosystem. And so the difference between having life turned on and turned off is a switch that's very finely tuned right around 0 degrees Celsius, right? 1 degree warmer and it's liquid and streams are flowing and lakes are filling up, and it's 1 degree cooler and everything is completely shut off. So it's really sensitive to climate. And I'll also point out that these ecosystems have not been disturbed by humans pretty much ever. There are no indigenous Antarcticans, right? So like if your canoe goes off course and you land in Antarctica, you're going extinct, dude. You're not going to survive that, right? There have no humans have ever lived there. So there haven't ever been any humans like pooping on stuff and peeing on things and plowing things and burning things down and messing up the environment, right? And so it's entirely pristine. The only disturbances that are there are human caused. They're either caused by us as scientists, or they're caused by the activities of humans that are acting very, very far away, right? So in that sense, right, like the burning of fossil fuels is changing climate. The actions of people thousands of miles away now are starting to have impacts on these ecosystems. So they're starting to change, and that's another good reason for us to study them. All right, so if you've ever played Jenga, I love this game, but I like to think of our ecosystem sort of like a Jenga game, where these Jenga blocks are like species in an ecosystem. And our system is so simple that we can experimentally remove or study how Mother Nature has experimentally removed species from an ecosystem and still have the diversity that's needed to support a functioning ecosystem, right? And so Antarctica is a great model, an analog of this Jenga game in terms of understanding the relationship between species biodiversity and the functioning of those species and functional ecosystems. All right, so yeah, there's, you see that, see my screen right there? Yeah, that's what 98.8% of Antarctica looks like right there. Not, yeah, for a biologist, not very exciting, I would say. But we work in the parts of Antarctica that look like this. You'll note that there are no grass or corn or rabbits or zebras or anything like that out there. Everything above ground is pretty much anything that's alive is in the soils, right? It's below the surface. There's a human in the lower right-hand corner there. Yeah, that's a human. Very cold, very dry, very windy environment. It's an analog of Mars. So a lot of Mars analog research is done here because it's similar in a lot of ways to the surface of Mars. The McMurdo Dry Valleys are ice-free, and they're ice-free because the transantarctic mountains over here on the left-hand side block the movement of the polar ice caps, the polar ice sheet, from moving across that part of the continent, leaving them ice-free. In order to get there, I leave from Utah, fly to San Francisco, and then to the North Island in New Zealand and the South Island in Christchurch. That's where we get all of our gear. We've got a big warehouse in Christchurch that's got all of our boots and gloves and coats and hats, and we ship all of our experimental cargo and stuff that we need down there for the field. And then when we get to Christchurch, we spend a day or two re-gearing and repacking our cargo. And then we fly down to Antarctica on the Air Force. The Air National Guard flies us down sometimes on these LC-130s that have skis. These are the only LC-30s outfitted like this in the world, and they allow us to do terrific science. It's a real advantage to us. After we get off the plane, we get to ride these super cool school buses with giant monster trucks. Ivan the Terror Bus. Yeah, these are giant tires. Look, there's some dude's head over here. You can see about how big these tires are. And before we can go out and do our research, we have to get trained to survive the harsh conditions in Antarctica. And so here's our teacher over here teaching us how to rescue someone who's fallen into a crevasse. We do all kinds of training like that before we go out. But once we get to the field, we stay in places like this or like this. There's my crib, nice crib. These are our living conditions. In this photograph, you can see my tent, my backpack, my sleep kit's got my sleeping bag and all that stuff in it. This looks like a water bottle, not a water bottle. That is a pee bottle, right? So you can't just go take a whiz behind a rock when you got to go, because remember what I said, this is a pristine environment. It's a natural laboratory. And so just like you wouldn't just go take a whiz in the corner of your lab or whatever, that'd be like messed up, right? Well, we don't do that in our laboratory either. It's very highly regulated by the National Science Foundation. And we have to keep track of every single disturbance we make. So every soil sample that I take has to get logged on a GPS unit and returned to the NSF at the end of the season so they can know all of the disturbance that we've made. So all the poops, all the peeps, everything that we make has to get packed out at the end of the season. The other thing I like to point out in this picture is that here in the background, you see the face of that large glacier. That's about 120 feet tall. And because you don't have any buildings or trees, you don't see, you don't have anything for perspective, right, to give you any way of measuring that, not just how big that is, but also how far away it might be. And sometimes that could be dangerous, right? Because you might say, hey, let's go hike over that glacier, and it turns out it's like 3 miles away or something. And weather can come up and get bad. So one of the challenges of working in Antarctica. All right, so what can Antarctic soil teach us? Well, if we know how ecosystems responded to climate warming in the past, we can better predict how they're going to respond in the future, right? So they're still very complicated, lots of ins, lots of outs, lots of what have yous. We don't know how the Antarctic soil ecosystems came to be structured the way that they do. And so that's an active part of our research program, is to understand this. So what we can understand how they've responded in the past. So Antarctica was not always super cold and frozen and covered with ice with nothing living above ground. During the Cretaceous, man, it wasn't too bad of a place. We had dinosaurs running around like this Cryolophosaurus, a theropod dinosaur. Yeah, not too bad of a spot to live. But then since the Neogene, then something happened, right? This happened and they all went extinct. So from the Neogene to the present, really, the organisms have had to respond to living in this kind of an environment. It's really harsh. Now, what are the charismatic megafauna of terrestrial Antarctica? For the past 20 million years, Antarctica has pretty much looked like this, right? Well, it turns out that there is a way to understand or to measure what a charismatic megafauna are. Right? So you can use this metric. I found this paper published in 2018, help us identify charismatic megafauna. These are the organisms that are beautiful or impressive or endangered or cute, dangerous or rare. So for example, giraffes are very beautiful apparently, and things like elephants are impressive. The blue whale is very endangered. The koala bear, really cute. Aw, yeah. Some things are enamored because they are dangerous, like the crocodile, or maybe because they're rare, right? And so I use these same criteria to evaluate the charismatic megafauna of Antarctica. Now, remember, we went from dinosaurs to something like having to respond to the cold. The birds figured it out. These penguins figured it out by, instead of flying through the air, they fly through the water, and they feed on krill. But unfortunately, these are not terrestrial animals. These are marine birds. These are seabirds. And when we find them in the dry valleys, they look like this. The dry valleys are no place for a penguin. It's too harsh of an environment. They suffer and quickly mummify. Now, there are seals in Antarctica. Oh, look how cute. Oh, megafauna. Yeah, megafauna, but they are not terrestrial animals, right? These are sea mammals, marine mammals. And so when we find them in Antarctica, they look like this. That there is no place in the dry valleys really for these seals. They for some reason wander up the valleys and end up dying and looking like this. I like to point out at this point, though, that if you were to just flip this cadaver, this mummy over, you would see that it's still soft fur on the bottom. It looks like it just died yesterday. This one's likely over 400 years old. These have been carbon dated by colleagues. And I point out that, like, the normal decay processes that would happen elsewhere on our planet just don't happen down here in Antarctica because everything freeze dries so quickly. So, like, if you dropped a banana behind the couch, you know, within a week or so, it would just be black slime because the bacteria and fungi start to break it down so rapidly. But the exposure that you see on this dead animal is not caused by bacteria or fungi. It's caused by ultraviolet radiation, pounding it, and then being sandblasted every year for hundreds of years. Yeah, cool, huh? So what's left? What really are the charismatic megafauna of continental Antarctica? Well, nematodes. Nematodes are the most abundant, widespread land animal of Antarctica. If you were kidnapped and then kicked out of an airplane flying over the continent and you landed and took a sample, it is most likely to contain this animal, the top of the dry valley's food chain, and a really important organism in nutrient cycling. So really important for ecosystem functioning in Antarctica. And I would argue that these animals are very beautiful. That is a beautiful animal. If you look at the sides of this, how it's pearled and how it reflects light, it's see-through like glass. That is a beautiful animal. Yeah. Other animals are like this tardigrade. So here's a tardigrade. His head's over here and his tail's over here and his little claws are gone. Aw. Well, here's an electron micrograph of a tardigrade. Yeah, everyone in my lab thinks these are super cute, right? So there's some charismatic megafauna with the cute factor. But I'd also say that this is a tardigrade egg, right? And so if you're a mom tardigrade trying to pass that thing, I'm like, oh my gosh, that also is impressive in a lot of ways. Yeah. We have rotifers in Antarctica. So here's the head of the rotifer, the anterior end. They have this corona that with these like flagella that whip around and create this current. You can see the stuff come flying down its gullet. So stuff comes flying in this direction. It stuffs down into its gullet and it gets chomped up here by these muncher things. Yeah, now that's a pretty cool animal. I would suggest that these rotifers are very impressive. Yeah. And then we also have mites. So these microarthropods are found here. They're not very commonly found, but you can find them. They are rare. And a couple of years ago, we found this columbella. Now, looking at that, you should have no problem finding it on the bottom of this rock. Look at that for a while. Yeah, if you can find that, you're actually pretty good. I know where it is, so I know where to look. But yeah, there it is right there. Oh, a little columbella on the bottom of that rock. If you look really close, there it is. Oh, yeah. Well, it turns out that this columbella is extremely rare. In fact, you can only find it on the equivalent of maybe 4 or 5 football fields in the central to southern transantarctic mountains. So only near the outlet of the Shackleton Glacier will you find this animal. Really cool animal. And there's that neat story in Scientific American. It just came out this month about that animal. I would say, maybe it's endangered, right? Just because of its range size is so tiny. So when I take a soil sample from a fairly rich site and I wash it through the screens and I extract those organisms from the soil and put them under a microscope, this is what it looks like under the microscope. So you can see that there are 3 different species of nematodes in here. There's at least 2 different species of tardigrades. And let me see if I can find some rotifers. Typically, I can find some rotifers, but I don't see one right offhand. But this is like a jungle. This is an Antarctic jungle that shows you the charismatic megafauna of continental Antarctica. All right, so that's all I wanted to talk about today. I just wanted to talk about the charismatic megafauna of continental Antarctica, give you a brief introduction into these organisms, and if we have time, then maybe I could ask some more questions about that.
Yeah, definitely. Well, bravo. Very entertaining. Very fun. And I learned a new term, muncher things.
Muncher things, yeah.
Muncher things, new tech.
Actually, that's not the technical term for them, but.
Sure. All right, we are open for questions. So go ahead, open up your mic and ask your question if you got something or a comment.
Hi, Byron. Sorry I was late. School stuff going on.
Yeah, I get it.
But it's really cool. What's the base of the Antarctic food chain in the soil then?
It's a good question. We're still working on that. As it turns out, we're still trying to close the carbon budget. So there's not enough contemporary primary productivity. There's not enough contemporary photosynthesis to run that ecosystem at the level that it's currently running. So I like to say it's running on fossil fuel right now. So the carbon, much of the carbon that's running these soils is carbon that was fixed thousands of years ago when the climate was a little bit, it was still cold climate, but the West Antarctic ice sheet had come in, filled the ends of those valleys, and it had blocked the ends of the valleys. And so these valley systems formed these really huge, these massive paleo lakes. And the paleo lakes were fairly productive. There's a lot of phytoplankton in those lakes. So they were able to capture a lot of energy from the sun and fix it by way of photosynthesis. And then as those lakes all dried down, then they left that carbon in the soil. And so the ecosystems now are using that old carbon in order to run. Now, having said that, I don't want to discount like the current primary productivity. There is some cyanobacteria that's able to photosynthesize, and we can find that in the soils on these hypoliths. It's kind of cool. These rocks that are light in color can allow sunlight to penetrate through the rocks, and so underneath the rock you'll have cyanobacteria that can survive there. And that's a carbon input into the ecosystem. But we're also starting to learn more about some of the microbes that are able to do autochemosynthetic carbon fixation as well. And we know that there are some that are there. The question is that a significant enough input to actually run this system at the level at which it's running? So, but we know there has to be more contemporary inputs than we think, because they should have burned all their fossil fuels by now if that was not the case. And they're clearly still working. So that's a great question, Bill.
I have a question.
Anybody? Yep, go ahead.
My name's Elaine, and I wanted to know, do you find any organisms in those streams that go from the glaciers to the lakes? Is there anything living in there?
There is. That's a great question, Elaine. There, on the tops of the glaciers, sometimes sediment will blow in, and that sediment will absorb solar radiation. It will melt down into the ice, and it forms like a little soda bottle of liquid water around it. And it forms a really cool bioreactor that we call cryoconites. And so algae can blow in there, and nutrients can blow in there, and those little reactors can grow and get big. Eventually they can melt out. As the glaciers melt, those flow into the streams. And so they can seed the streams with biota and also with nutrients. And so those organisms get into the streams. The streams and the moats are the most ecologically productive, the most active components of this ecosystem. So in terms of primary productivity, most of the contemporary productivity that we measure occurs in the streams and in the moats of the lakes. Yeah, great question. And the most biologically diverse, at least microbially, are the streams. So the streams have the greatest amount of microbial diversity of all the landforms. So they're more diverse than the glaciers or the soils or the lakes. They tend to be like a catch-all. And it kind of makes sense. We have some experiments to test this hypothesis, but if you look at the topology of the streams across the landscape, you know, they set down a little bit low on the landscape. And so as the winds blow stuff, those stream channels just catch it. So the stream channels catch biota that's blowing around, they catch nutrients that are blowing around. So it kind of makes sense that those would be really diverse parts of our ecosystem. Yeah, great question.
Anyone else?
I was thinking about the scale when you showed that assemblage photo of the tardigrades and rotifer. Well, I don't know if there's a rotifer and the nematodes.
Uh-huh.
What volume of soil was that from?
So that's from about 100 grams of soil, which typically is a little bit less than 100 cc's of soil. So about 100 grams of soil, but we have to, obviously in order to compare these, what we do is we calculate what the soil moisture is, so the gravimetric soil moisture, so that we can compare like the number of animals per dry mass per kilogram of soil. And so in the lab, you're going to get to do a whole bunch of this, Bill, till you probably can't stand it. But yeah, we'll take these samples and we'll put them into tins at 100 grams each, but then we have to dry the tins in an oven to measure out what the gravimetric soil moisture was. But soil moisture ends up being a very important variable that we have to look at because it's a good predictor of where you find life, right? And activity in the soils. Yeah.
Hey Byron, this is Scott.
Hey Scott.
Hey man, long time.
It's been too long, dude.
Hey, I was wondering, have you guys ever looked at like depth profiles and how far down these communities go?
That's a great question, and we just started talking about that again as part of our team. We're interested in this. There were some early studies that showed that about 90% of all the biota exist in the top 5 centimeters of soil. We, for our studies, we go to the top 10 centimeters of soil. But in some places, you can go down to even like 60 centimeters before you hit the ice cemented concrete. And even having said that, I've gone places where I'll hit that permafrost, that solid ice barrier that at maybe even 10 centimeters. And I could use an ice axe and I can like chip it out. It's like concrete. And I can take it back to the lab, and when I thaw it out, there's animals that are living in that stuff. You know, they're alive. It's kind of crazy. So I think that the biota by depth, we should probably look at that again to better understand it. But for the most part, like I said, most of the biota is in the top 5 centimeters or so. And across the landscape, you get down to about 15 to 20 centimeters, and then you're hitting that ice cemented concrete where it's permafrost, like that's been frozen for thousands of years likely.
Byron, I have a question. This is Kara up here.
Hi, Kara.
But you're in the LTER region, and I'm just wondering, over the time that you've been collecting these samples, have you seen any changes in like the biodiversity in terms of species number or in terms of maybe population numbers? Like, as you know, we're reading all of this about, warming trends, things like that. Are you seeing changes that you can identify yet, or is that still not quite enough data for that?
Yeah, great question. So if I were to just sort of look at raw numbers on my spreadsheet, or people ask me like anecdotally, are there any major changes that I could put my finger on? I would initially say no, because there's so much interannual variability. There is no such thing as like an average year. Every year is kind of different and weird down there. So it's not until we start like studying our data with, with some good statistical tools that these trends start to show up. And I like to point out to my students that had we only gone there for 1 year or 2 years or 3 years, we would have told a different story drawing trend lines every single time. But now that we've got 30 plus years of data, now we can make inferences that are a lot more meaningful. And yes, so that the answer is that it appears that our ecosystem is getting a little bit warmer and a little bit wetter, but not on a trend line like what you see in the Arctic so much. What we're starting to see is the frequency and magnitude of these pulse warming events is starting to change, right? So that these pulse warming summers are becoming more frequent, and the magnitude of that pulse warming is becoming is increasing. And that's led to changes in the community structure of these soil organisms across the landscape. So there's an animal that really likes it cold and dry and salty, and in response to these changes, that animal's abundance is starting to decrease, and its distribution across the landscape is also starting to decrease. And these other animals that are more mesic, they like it wetter, they like it a little bit warmer, they're starting to expand their ranges across the landscape. So yeah, we are starting to see this, but it's again, you need to approach it with statistics. It's not like someplace in the Arctic, you're just like, wow, this island wasn't here last year, and here it is. Like some of those changes are massive and major and very rapid. Whereas ours, we're still at the very front end of trying to of detecting anthropogenic-driven changes. And that is very different than what you would see on the peninsula, right? On the peninsula, you're seeing really dramatic changes that are anthropogenically driven, climate types of changes. But we're just so far interior to that, that we're buffered by the circumpolar vortex and some other physical things that are preventing us from feeling those effects quite as profoundly as in the more northern latitudes.
Great, thank you.
Sure.
No one else?
I have another question. So like a lot of the other LTER experiments, they can kind of compare locations. Is there any talk about expanding to another Antarctic location to kind of compare the research you guys have had?
That's a great question. We've always wanted to be able to compare our work with the site that's at Palmer, for example. Most of the work that's done at Palmer is marine based, and we've always wondered, well, what about the soil ecosystems that are in and around Palmer? Well, there's not a whole lot of soil in and around Palmer, for one thing. But we have, we wrote a paper, a series of papers actually in Bioscience a few years ago, where we compared some of our pulse climate events and the effects that they had on our ecosystem with what the Palmer LTER was detecting and the effects that they had on their ecosystem. And it was really super cool. Like I think, like I study worms, they study penguins. What is there to talk about, right? And you get us together in the same room, and Bill Fraser's showing his penguin data, and I'm like, hey, that looks just like my worm data. Like, what's going on, right? And so then we start talking at that level, and we were able to write some really compelling papers about the differences between the two, and because they were very different. This unifying effect, right? This one thing that happened in Antarctica sort of affected our ecosystem in one way, and it affected theirs in another way. But both ecosystems had these pronounced responses to this pulse warming event, this really weird thing that happened with the southern annual mode. And so in that sense, yeah, we're always trying to connect our research to the research that's been done at other sites. And so there's some logical connections. We have a lot of lakes in our ecosystem, so we collaborate with folks that are studying lakes at the other LTER sites, and there's a lot of lake work that's done out there. And we're a polar desert, right? And so we have lots of analogs with like the Sevieta LTER site, the Sevieta Desert, and the Jornada deserts. And so, yeah, we find connections between our site and other sites at a lot of different levels.
Can you explain the pulse warming event a little bit more? Like, is that like just over one season, 1 week, months, year?
Yeah, it's usually
look like?
yeah, I can see how that could be confusing. So usually this takes place just over like a week or two. And what happens is you get clear skies, you get the sun, and it also involves the ozone hole in a complicated way. But in general, what's happening is we're getting increased insolation, so increased solar radiation over our site without any clouds, right? And it doesn't have to change ambient temperature very much, right? So the temperature could be like, 20 degrees Fahrenheit, go to 28 degrees Fahrenheit or whatever. But what's really happening is the surface of our ecosystem is absorbing a lot of radiation. And so that causes the glaciers to melt and melt very rapidly, which means our little trickling streams turn into raging rivers, and the lakes can fill up as much as like a meter, like that's one of the cool things about the lakes in this ecosystem is that they're all closed basin lakes, so like bathtubs, right? And they integrate climate perfectly because when it's warm, glaciers melt, lake levels rise, right? When it's cold, you get a lot of ablation and sublimation, and so the lake levels drop. And so we've got a nice historical record of stream flow and glacial mass balance and lake level rise that it tells a really nice climate story. And the story that it's telling is that climate's changing, but we get these pulse events, like in 1 or 2 weeks, you can get enough melt to undo like 10 years of a cold spell, for example. So you can have lake dry down for 10 years, and in like 2-3 weeks, boom, it's like 10 years worth of melt all at once. So that's what we mean by these pulse events, and the frequency and magnitude of these pulse events changing. And that's probably the big change that we're starting to see down there.
Thank you. Anybody else with questions?
I have one for Judy and Janet and Byron. At what point are you guys and ladies going to be getting a little bit nervous about next year's science down to the ice and sending people out?
Well, I'll take that to start with because I'm not nervous.
Go ahead.
Yeah, so we're hopeful. We plan like crazy. We know that planning is super important and super helpful. In the end, we all know that all of our plans are completely worthless. And so like our lead PI, Mike Gusev, likes to quote Mike Tyson. He says, Mike Tyson apparently said at one point, yeah, everybody has a plan till you get punched in the face. And so we make plans, and we plan for this season. We've already planned all the stuff that we're going to do. We're working really hard to implement all those plans. And if something happens, something happens. But we're very optimistic that we'll be able to complete our whole season and do it successfully.
Yeah, what he said. All right. Anybody else? Questions, thoughts? All right. Well, thanks, Byron. It was really interesting. I learned a lot, actually. And yeah, thanks everybody for joining us. We will share the archive and post it online when it's done and ready. And yeah, we look forward to seeing you all again.